to catch this. My name is Trudy Ray. It is currently not clear how long SARS-CoV-2 vaccine-induced immunity lasts. The gold standard for determining the efficacy of a vaccine is the challenge study, which involves intentionally infecting immunized subjects with the pathogen against which they were immunized. However, because it is unethical to deliberately infect humans with pathogens that cause serious morbidity and mortality, and SARS-CoV-2 definitely falls into this category, such studies are typically done in non-human primates. In this video, I will describe one such study. A recent preprint by Kizzmekia Corbett and others describes experiments done to assess the efficacy of Moderna's SARS-CoV-2 Spikevax vaccine in rhesus macaques 1 year after vaccination. The authors immunized 8 animals with 2 doses of Spikevax at 4-week intervals, which is the same regimen given to humans, and then they collected blood samples, nasal swabs, and lung wash samples at various time points over the course of the following year. The macaques were then challenged with the SARS-CoV-2 Delta variant virus at 49 weeks, which is about a year, and more samples were collected at different time points after challenge. The investigators collected blood samples at 6, 24, and 48 weeks post-vaccination, and they analyzed IgG antibodies in these samples. Now remember, IgG antibodies are mostly blood resident and provide the majority of antibody-based immunity against invading pathogens. The IgG antibodies were tested for their ability to bind the receptor binding domains of 3 different viruses: ancestral SARS-CoV-2, the Delta variant, and the Beta variant. Ancestral virus is the virus that first emerged before it had a chance to mutate. And this virus had the exact spike protein antigen encoded in the vaccine. So basically it matched the vaccine. And the Delta and Beta variants had various changes in their spike proteins. The investigators found that IgG levels were highest at 6 weeks after vaccination for all 3 viruses, and then they declined rapidly between 6 weeks and 24 weeks, and more slowly between 24 weeks and 48 weeks. Most IgG detected at 6 weeks bound ancestral virus, with 5.4-fold fewer IgG molecules binding the Delta variant and 8-fold fewer IgG molecules binding the Beta variant, in comparison to IgG specific to the ancestral virus. However, when Delta and Beta variant specific IgG antibodies were tested for their ability to block binding between SARS-CoV-2 and its cognate ACE2 receptor, so now we're no longer talking about antibodies just being able to bind their target virus, we're talking about their ability to get in the way and prevent the virus from binding its receptor to get into the cell. They inhibited almost 100% of binding of both Delta and Beta variant viruses, suggesting that the antibodies were still functional in preventing infection, in spite of their diminished quantity. The ability of blood-resident IgG antibodies to neutralize the 3 respective viruses followed a similar trend, with a gradual decline in neutralizing activity against all viruses by 48 weeks post-vaccination. Interestingly, even though the quantity of total binding and neutralizing antibodies targeting the Delta variant decreased over time, the number of antibodies targeting regions associated with neutralization increased. In addition, the binding avidity of antibodies to ancestral virus increased significantly between week 6 and 24, and remained steady through week 48 post-vaccination. In contrast to affinity, which measures the strength of the binding interaction between antigen and antibody at a single binding site, avidity measures the total binding strength of an antibody at every binding site. These 2 shifts, the increase in the number of antibodies binding targets associated with neutralization and the increase in antibody avidity over time in spite of a decrease in total antibody levels, are suggestive of a maturing immune response that is more focused on viral regions of high immunological relevance. I would like to mention that the regions associated with neutralization are outside of areas where the variant viruses have accumulated changes in the spike protein. And this further implies that Spikevax and other SARS-CoV-2 vaccines are just as effective against viral variants as they are against ancestral virus. Next, the authors analyzed lung wash samples and nasal swabs for Delta-binding IgG and IgA antibodies. IgA antibodies are predominantly found in mucous membranes and their fluids, where they protect against invasion by inhaled and ingested pathogens. IgG kinetics in the lung were similar to those observed in the blood. Both binding and neutralizing IgG to all three viruses were highest at 6 weeks after vaccination and decreased steadily over time until they were indistinguishable from those observed in unvaccinated animals. In contrast, IgG levels in the nose increased through week 25, plateaued, and remained stable through week 42 post-vaccination. IgA levels in the lung were highest at week 6 post-vaccination, but decreased to levels similar to those observed in unvaccinated animals by week 24. IgA levels in the nose were similar to those in unvaccinated animals at all time points. These results suggest that although SARS-CoV-2 vaccination may not induce a detectable mucosal immune response in the nose, it does induce good initial mucosal immunity in the lung, which is typically the site of severe COVID-19. This immunological difference between the lung and the nose might also explain why SARS-CoV-2 vaccines are more effective at preventing severe disease than at preventing infection. The authors also analyzed blood samples from vaccinated animals for the presence of SARS-CoV-2 specific memory B cells, which can quickly make spike-specific antibodies upon a subsequent exposure to SARS-CoV-2. At week 6 post-vaccination, about 0.14% of all memory B cells were specific to the ancestral virus, and about 0.09% were specific for the Delta variant. In comparison, about 2.5% of all memory B cells were specific for both the ancestral virus and the Delta variant, and this high proportion of dual binding to single binding cells remained constant through week 42 post-vaccination. In other words, the memory B cell response was good, but it was better for B cells specific for both viruses, and it was more durable. To see whether these vaccine-induced immune parameters are protective after viral challenge, the authors infected the animals with Delta variant virus at 49 weeks after the initial immunization. So this is almost a year later. Basically, they wanted to see how good that immunity was at the 1-year mark. Lung washes and nasal swabs were collected on days 2, 4, 7, and 14 after challenge to monitor viral replication. On day 2 after challenge, vaccinated animals had about 11-fold fewer viral RNA copies per milliliter in their lungs than unvaccinated animals, and these RNA levels declined rapidly over the following days. In contrast, viral RNA levels in unvaccinated animals remained significantly elevated through day 7 post-infection. Viral RNA levels in the nose followed a similar trend. However, their decline in vaccinated animals was not as dramatic as that observed in the lung. Basically, this suggested that vaccinated animals may have had virus in the lung for shorter periods of time than unvaccinated animals, which is not surprising. Antibodies to all three viruses in the lungs of vaccinated animals were significantly higher on day 4 after challenge than at week 42 after immunization, suggesting that memory B cell responses to infection were quick and robust. Viral challenge after vaccination also induced both T helper cells, which stimulate B cells to make antibodies, and cytotoxic T cells, which kill virus-infected cells. Analysis of lung tissue also revealed that vaccination prevented lung pathology and protected the lower respiratory tract from severe inflammation after infection. To me, the most interesting observation in this study relates to whether vaccinated individuals who become infected replicate and transmit virus to others. Basically, if you are vaccinated, does that prevent you from transmitting virus if you become infected? When the authors analyzed lung wash samples for T cells specific for the SARS-CoV-2 N protein, which is not encoded in the Spikevax vaccine, they only found these cells in unvaccinated animals. The viral N protein can only come from infection because the vaccine does not contain this antigen. This suggests that even though it had been 1 year since vaccination, immunized animals that were then infected did not replicate the challenge virus to a sufficient extent to produce T cells specific for the N protein, a response that would only be elicited by actual infection with whole virus. In other words, the memory response to the SARS-CoV-2 spike protein induced by the vaccine eliminated incoming virus so quickly that the immune system had no chance to mount a response to the viral N protein from the challenge virus. presumably because the virus was cleared so quickly. In summary, vaccinated animals appear to be better protected from severe disease and to clear virus faster than unvaccinated animals. This result aligns with data published in a previous preprint, which showed that viral RNA levels in Delta variant infected people who had been vaccinated prior to infection declined more rapidly than in people who were not vaccinated. And although monkeys are not human, previous studies assessing the protective efficacy of Spikevax have shown that rhesus macaques are reliably predictive of outcomes in humans, making them a great model for determining the effects of waning antibody levels on long-term protection against SARS-CoV-2 infection. I think these results are very promising. Thanks again for listening. My name is Trudy Ray. I'll see you next time on Catch This.